My talk is titled 50 parameter CyTOF intracellular panel reveals distinct functional T cells, but I'm going to walk you through the whole journey, how we got there from smaller panels to larger, and how we assess different avenues of methodologies to reach the point where we are now. And I wanted to start historically. So it's been about 40 years now when the existence of different T cell lineages has been first discovered, and when it was found that there's the shape of memory T cells can be defined through the environment that they're maturing in. So the T cells can differentiate into Th1 or Th2 functional phenotypes depending on the cytokine environment and other microenvironment factors around them. So if they're subjected to IL-2, the IL-12, they would differentiate in Th1 arm, and IL-4 to Th2 arm. I mean, this is all like history, everybody knows that now. But that was groundbreaking that we have different flavors of T cells, and they would produce different cytokines that would eventually end up crucial for different immune aspects of immunity. So the Th1 arm was first implicated to be part of the anti-tumor immunity and antiviral clearance, and also the Th2 arm is evolutionary, is part of our defense against worm infections. And also we have all the aspects of autoimmunity coming up from the disbalance of these two arms, with Th1 leading to one side of the autoimmunity conditions and the Th2 arm leading to allergies and asthma. So now we're going back to 2025, and we know that we have way more flavors of T cells, right? So we have this type 1 and type 2 historically that we know, and then we have Th17 cells, we have type 22, we have T follicular cells, and yada yada. I'm sure this is not a full list. So the cells are defined through the transcription factors that govern their transcriptional profiles and programs, and also their characteristic cytokines that they make. So we can fingerprint T cells based on the cytokines they produce, and that's the ultimate way to define their function, right? So if we're talking about the phases of the immune response to pathogen, so we have the phase 1 when the antigen is encountered, there is inflammatory response and the active clearance of the pathogen, and then we go to phase 2 when there's a ceasing of inflammation and curbing the cytotoxic responses that become actually damaging to the host. And then we have phase 3 that is restoring tissue homeostasis. And at all these 3 stages, the cytokine profiles are different, and the cytokines that the cells make are really defining what's going on in the tissue. Not even transcription factors, but the cytokines, because that's the function that's exerted, right? So if we look at the surface markers, the classic phenotyping concept of surface markers, it's only the tip of the iceberg, and the actual function can be only defined if we look at what's happening inside the cell, so the intracellular functional markers. And I mean, historically, we were interested in the cytokine profiles, but we also have all kinds of other aspects that people look at: the metabolic programs, the transcription of factors, the signal transduction and phospho cascades. So all this kind of analytes that we can define to figure out what kind of complex program is happening in the specific T cell and in the environment of the multiple T cells that are invading the tissue or they're circulating in the blood. So this would give us the understanding of the real link between the surface phenotype that is often commonly addressed by other methods and the actual cellular action, right? So for mechanistic research, this is crucial to have this defined, okay? And it's different from looking at, for example, we have all the single-cell RNA-seq data now, but that's very remote from the actual exerted function. So we're going at the end of action, the cytokine that's getting out of the cell and exerting the function. So we started with a very modest request. We didn't want much, right? So we wanted to have a panel where we could define some major cytokines. So it's not an excessive list that you can, any cytokine you can think about. So classic Th1s, classic Th2s, and cytokines that are made by multiple cells that define the pro and anti-inflammatory responses. So, so we made a small list, and my major methodology at that time that I utilized, and I utilize up to this day, was flow cytometry. I was like, yeah, we will make a flow cytometry panel. I'm fully confident we can do this. So we can go not really super big, maybe 30 colors, and measure the cytokines along with the surface markers. Why can be this difficult, right? So, and the result was that certain cytokines worked beautifully, but some did not, okay? And I mean, trust me, we know how to troubleshoot flow cytometry panels. I have a reputation of knowing how to do that, and there's a big community that can help me, so I was like, okay, we're gonna spend some time and figure it out. And it's been about a year that we spent to figure it out. And unfortunately, although we knew that the cells are actually making the cytokines, so we measured cytokines from the same samples by ELISpot, for example, like an orthogonal technique, so we knew that we have cytokines made. We couldn't success in certain cytokines detection. Some were marginal, and some were really beautiful. And we did all the usual things, change the colors, deconstructed the panels to 3 colors, changed the fluorochromes, all the things you can imagine, right? And there were just some stubborn analytes that didn't work, okay? And unfortunately, we're not alone in that. If I go back to the literature, I can see that some cytokines, and they're spoken through in this slide, right? They're not really measured well in the data that is produced by other labs. And I want you to look at this and think, when you see the paper that talks about cytokines measured from the cells, which ones do you usually see? It would be the ones in bold here, okay? It will never be IL-5 or IL-13, okay? So this forms this, let's look under the street lamp mentality where the things that are easy to measure are constantly measured again and again and again, okay? And if you think about PBMCs from healthy donors that we always use for troubleshooting any panels, the Th2 cytokines may be a very small fraction of what we want to measure. But if we go out of that context to real life context, this becomes a real problem because we're only seeing a pro-inflammatory arm of the immune response. We're only seeing phase 1 cytokines detected, and we carry this bias into these studies where we're looking at certain aspects of disease, or we're phenotyping cell therapy products, and we still measure interferon gamma and TNF-alpha, right? So this is when we were like, okay, we weren't actually, people from the mass cytometry field found this moment to lure us into mass cytometry world. So we built a panel with mass cytometry with the same set of markers, I mean, more or less, maybe a few were different. And I mean, everything got detected beautifully. And I was shocked because, okay, these experiments were not aligned 100%, but still we used the same clones. I couldn't understand why this not works as expected. And we wanted to do a clear-cut study, so we did back-to-back analysis. So we made a simple, small flow cytometry panel, only 11, 12 colors, and with lineage and with cytokines, a handful of cytokines. So made sure that there's minimal spectral overlap. I mean, we're all using spectral cytometry. 12 colors is nothing, okay? We used identical antibody clones for CyTOF and fluorescent cytometry. We tried to mimic the same experience as people would have in the real life. So we would basically pull the antibodies that are readily available by commercial sources for flow cytometry, and for mass cytometry, we would do some custom conjugations to match this. So this would be the real life experience for a user who was trying to do that. And we had the same culture split into 2 arms. So there's no question that oh, maybe you process the sample different, stimulated different. It's the same culture. We used the same set of buffers. Everything was same. And basically, what we found is that, as you can expect, and this data, we've shown it around, if you've been at the commercial tutorial last year, Jennifer was showing this slide, so I'm going to go through this really quick. So the ones that are, expectedly working, they work both platforms had no issues with them, right? But as we go to the ones that were like problematic, so the only one that came out of our extensive troubleshooting with improvement for flow cytometry was IL-5. We finally got it working. But if we look at, for example, IL-10, so, and again, we deconstructed this to like a 3-color panel. It's not the flow, like any flow disturbances that you can think about, right? So here we can see the pattern that is pretty typical for other things that we were troubleshooting, where the background is so strong that if you titrate the reagent down, you end up still with the background and the signal is not there. So we can get a very clean signal for CyTOF. It's not a lot of cells because this is healthy PBMCs, but it's very distinct compared to the unstimulated conditions. And with the flow cytometry, we just see the background that we cannot get rid of. And with the IL-13, we see a different pattern. That was the most striking, I think, result that we ever had. So again and again, we see it. IL-13 is not detectable by flow cytometry in the hands of like 10 labs that I checked with. I mean, you can see 5 OMIPs that have IL-13, and the data look really poor. It's hard. It's hard. You cannot really see it no matter what color you use. I mean, with CyTOF, we know that there are buckets of IL-13 in the supernatant, and we can detect the cells easily, one again and again and again. We've seen it so many times now, it doesn't fail to impress me every time I see this. So we have a phosphorylation and transcription factor panels. I'm not going to talk about them now, but we will have a talk on the last day of CyTO in the parallel session where we go through this in more detail. And also we have a paper uploaded to bioRxiv, which is now chewing it for 24 hours, so I cannot put a QR code. But if you take a picture of the slide and then look it up, like maybe tomorrow, it will show up. Or come to the talk, I will have a QR code then. So why does this happen? Honestly, we don't have a hard proof why. So we got rid of all the possible causes of failure of one technology versus another, right? So we got rid of spectral overlap as much as we could. The autofluorescence is curbed down. It's all T cells. We still have some scenarios why this could be happening. So we still have the background too high in flow cytometry. So maybe the signal is so dim that we cannot really get over that background. And also the big thing is that the size of the reagent is not same. So the antibody conjugated to the fluorochrome is way larger than the antibody conjugated to the metal, right? The molecule size is different. Also the hydrophobic cloud around the conjugate that you have often is big. So the penetration of the reagent into the tissue into the cell is much more difficult for flow cytometry reagents. And although we really don't know why one cytokine works really well and the other cytokine doesn't, but we don't know much about the complexity of the intracellular structures, how they actually partition the cytokine in the cell, especially given that it's not a physiological situation. We add Brefeldin and monensin there, right? So something prevents maybe the antibody reaching the target, but it doesn't prevent it from non-specific binding and creating the background labeling, okay? So now with all that, we are like, okay, the world is our oyster. We know that this works. We want to do a big panel for fingerprinting cytokines. So there is a poster, so you don't need to squint to look at all the stuff that I'm showing now. You can go to the poster and get as close as you want, or you can download this poster. So the poster will be presented, it's 171, I think it's on Monday. And Laura Polanco, who was doing a lot of this work, is here. So you can corner her and ask her grill her for questions. So she's Jennifer's graduate student. So we put lineage markers for like surface to define common lineages. This can be easily, flexible for specific lineage that you want. I mean, it's a pretty exhaustive list, but if you want something extra, you can always add it there. It's very T-cell heavy, as you can guess, because we're T-cell people. So, and we put 24 cytokines in there. So name your favorite cytokine. There is a good chance that it's there. I mean, a T-cell cytokine. But not only T-cells, there are cytokines that are made by myeloid cells and B cells, and we have actually a few like B-cell myeloid cell markers in the panel. So, and a lot of them we put there because we don't even know what they do, because there's no literature, because everybody is measuring interferon gamma and IL-4. Okay, so yeah, so we were absolutely open to exploratory findings. So the workflow is like pretty standard. We're not doing flow cytometry back-to-back anymore. It's not possible to do flow cytometry for this. We know that. So we go straight mass cytometry. So when we do phenotyping, I mean, we get our usual, PBMC map of all possible populations there. That's not pretty trivial, right? So specifically for this talk, I wanted to pull out the memory, the T cell memory, because this is the cells that we expect to have biologically relevant cytokines produced from. Because, I mean, also our stimulations are kind of artificial. They show what the panel is capable of, but biologically relevant is just the cytokines that are produced by memory cells, right? So I pulled out the clusters that correspond to CD45 RO positive T cells. And I mean, we clustered them. There's this map that everybody loves to look at, right, and squint around. So yeah, please go to the poster. But I just wanted to point to like a couple of interesting things here. So we can obviously detect Th1, Th2 lineages. We can detect some cytokines that are only produced by myeloid cells, so we don't really see them in T cell lineages, but they're not common to see in other phenotyping projects. So if we see some T cells suddenly expressing these cytokines, we're ready, okay? So, and I mean, we were able, because this is way past the time when I was saying, oh, mass cytometry is not good for us because we're interested in iNKT cells, and there are so few that we need to record so many events, and mass cytometry can't allow us to do it unless we spend our life chained to the old, CyTOF 2 unit. So that's not true anymore. And I mean, I have the most recent data set that I saw. I saw my iNKTs there because we easily could collect, 500,000, 1 million events from the sample, and then I concatenated several samples, and I could see the iNKT population just nice and clear there. So that's actually one of the weaknesses of mass cytometry, I think, is like going away with like the limitation of the cell numbers that you can assess from one panel, okay? So we can confirm all the usual things that you expect from that panel. I mean, we can see all the Th1, Th2 lineages. We can see the Th2 cytokines beautifully expressed. As I said, I'm always happy to see IL-5 and IL-13 co-expressed in the cell. And I mean, we have interesting complexity of Th17 cytokines presented there. So we have IL-17, IL-22, IL-21. There are patterns that are, I think, worth looking at. Again, go to the poster, squint, and look at them. And one thing I'm really excited about to show, and that's the last thing I'm going to show, is that we have the Treg enriched area. So I just boxed it on the plot here. So this is the OPSNI map just blown up so that you can see it. So CD27 low, CD25 high cells. And that's the only place on the map where I can actually see TGF-beta. And TGF-beta is like, I mean, I love to see it. I have never been able to see it with flow cytometry. It looks so good. So this is my TGF-beta enriched area of the map. And I can see that there's also some Tregs that are making IL-10, and IL-4 and IL-17 have a very expected pattern of expression. So this looks to me a very nice confirmation of how our panel is performing for the populations that are defined by current knowledge of immunology, right? So just to conclude, so what I want to say is that our panel is ready to be used for fingerprinting PBMCs and cell therapy products. All their functional capacities in the context of inflammation or cancer or whatnot. And what's important is that this flexibility of making this panel and approaching any troubleshooting of the panel it allows us to apply it to the context where we have unknown cytokine production patterns. And I want to show you, like, this is a small example. So we looked at the iPSC-derived CD4 cells. That is a super hot thing in our institution. We have a lab that is doing this, and they're the first lab in the world who was able to do that. So, and these are CD4 cells. They look like CD4 cells. They are amazing. But when we fingerprinted them, a good proportion of them made IL-8 in buckets, okay? So if it was a flow panel, I would never put the IL-8 on the channel that would have issues with, like, T cell markers, because I would say, well, it would be on myeloid, so I'll design my panel around it. And it would be a curveball that panel would not survive. Now, we had here, like, we could detected perfectly. We wanted to adjust a few metals, but it's so easy to fix it, right? So when we have a confirmatory study where we know exactly what to expect in terms of expression, I'm so happy to use flow cytometry, because that, like the fluorescent cytometry, because it produces the results that I expect to see. But once I go to the exploratory analysis and unknown contexts, the fact that with CyTOF I don't have the big interaction between different analytes, and I get more independent measurement of each analyte, is absolutely golden. That's the reason why I would go with exploratory analysis with this platform. So we're super excited to do this all. Just to disclose, this part of this research was funded by Standard BioTools. And I wanted to say my acknowledgements. So most of the work here was split between the two labs. So the lab of Jennifer Snyder-Capioni at BU, with whom we work in tandem. So I'm a director of the flow cytometry core facility at BU, and also assistant professor of pathology and laboratory medicine. So I do immunology research, and Jen's lab is focused on immunology research in studying chronic inflammation and different aspects of innate immunity related to chronic inflammatory conditions. And the other hand of this research was done in Standard BioTools labs in Toronto. So we work with a team of Christine Loke, who's not here, but you all know her, and her team was doing a lot of the wet lab experiments related to this.